Physiology of digestion. Digestion of carbohydrates. First step is the digestion of carbohydrates in the buccal cavity. The buccal cavity has saliva which contains salivary amylase. Salivary amylase gets activated by the chloride ion present in saliva. Salivary amylase breaks down starch and glycogen into dextrin and then into double sugar like maltose, isomaltose and small dextrins called limit dextrin. Stomach. The salivary amylase activity stops in stomach by the action of hydrochloric acid. In small intestine, pancreatic juice is received that contains pancreatic amylase having the action similar to salivary amylase. Intestinal juice is also received in the small intestine. It contains several enzymes like intestinal amylase which has action similar to salivary amylase, maltase which breaks down maltose, isomaltase that breaks down isomaltose, limit dextrinase that breaks down limit dextrin, sucrase that breaks down sucrose and lactase that breaks down lactose to simple sugars. Digestion of fats. In buccal cavity, there is no specific enzyme present for the digestion of fat. In stomach, there is no emulsifying agent present, but the gastric juice has gastric lipase which converts some fats into monoglycerides and fatty acids. The important step of digestion of fats takes place in the small intestine, as the small intestine receives the bile from liver that has the bile salts which acts as the emulsifying agent, breaking down the large globules of fats into smaller one, thus making the action of lipase enzyme faster and easier. The small intestine receives pancreatic juice. Pancreatic juice has pancreatic lipase, which is a main fat digesting enzyme. It breaks down fat at various stages. Lipase enzyme separates one fatty acid molecule from the triglyceride, changing it into diglyceride, and then the diglyceride is again broken down by the removal of fatty acid and changes to monoglyceride. At the third stage, monoglyceride is broken down, separating the fatty acid molecule, and a glycerol molecule is left out. The small intestine also receives intestinal juice which has intestinal lipase. This enzyme occurs mainly in the intestinal epithelial cells and only a small amount is released in the intestinal juice. The lipase found in intestinal juice breaks down triglyceride, diglyceride and monoglyceride to fatty acid and glycerol like the action of pancreatic lipase. The intestinal lipase present in the intestinal epithelial cell hydrolyzes the absorbed triglyceride and diglyceride to monoglyceride and fatty acids. The fatty acids, glycerol and monoglycerols are the end products of fat digestion. Thank you so much.